Good evening, all of you. Welcome to the 13th webinar of the Thursday webinar series of IEEE Malabar Subsection. So, as all of you are aware, from June 1st onwards, IEEE Malabar Subsection is organizing a webinar on every week. So, this is the 13th webinar in this series. So, the coming week, we are having a speaker from IIT Bombay, and there are many interesting topics and speakers who have signed up for this webinar series. So, today we have Dr. Gopakumar from Computer Science and Engineering Department of NIT Calicut. He's a person who is working in the bioinformatics area. So, today he is going to talk on a very interesting topic. So, I'm handing over mic to Dr. Taslima for anchoring as well as telling all the rules of attending this webinar. I hope this will be a very interesting webinar and everyone will get benefited by this talk. Over to Dr. Taslima.
Thank you, sir. It's been a great privilege for me here today with this 13th webinar, that is webinar on our IEEE webinar series organized by this IEEE Malabar Subsection in collaboration with the IEEE Malabar Hub. So today's topic is with the computers and coronavirus, and we are blessed with the presence of the eminent resource person, Dr. G. Gopakumar, currently working as an assistant professor in the Department of Computer Science and Engineering, NIT Calicut. And my duty is to introduce the speaker for the audience. So that is about our speaker of the day. He has completed his PhD in bioinformatics from the Cochin University of Science and Technology. And he is having 5 ongoing research students working under his guidance, and he has already completed 2 research supervision as a co-guide. And he is having in his credit 5 SCA indexed journal and 4 Scopus indexed journal publications, and he has attended 10 international conferences in various places. He has been published one of the paper like a GPU-based virtual screening tool using random forest classifier in the Journal of Cheminformatics. That was an SCA indexed journal, and he has having many publications as a science citation indexed publication in his credit. And his under his supervision, 2 research scholars are completed their research. That is GPU-based virtual screening techniques for faster drug discovery and seed-based DNA sequence computation with a tandem repeat elimination. He's already having an experience with organizing different consultancy program, faculty development program, and also he has organized different workshops as an organizer, coordinator. So he's already having some administrative responsibilities, which includes as a department level purchase coordinator, department level TechHip coordinator, faculty in charge of big data and bioinformatics laboratory, faculty in charge of campus networking center, and a member in the Institute of Website Maintenance team. So considering all his experiences, we have got a very good resource person who can speak more about what is the relation between these computers and coronavirus, and definitely all the participants will be getting a very better session today. So thank you so much, sir, for being with us today for this evening session. Over to Dr. G. Gopakumar, sir. Thank you.
Yeah, thank you, Taslima, for your kind introduction. And thank you, Madhumar sir, for kindly inviting me to this very prestigious IEEE webinar series, which I consider is a very unique initiative from IEEE Malabar section. So it's really a commendable one, actually. So I am actually fortunate to have to present a session today. So I thank the organizers for considering me and giving an opportunity to me. So today, so I will share my screen. So I think
Yeah, it's visible, sir, but
visible.
presentation.
Okay, fine. So is it visible?
Yes, sir. Okay, fine.
So fine.
Sir, shall I just some instruction to the participants? I request
Yeah,
all the
please.
participants to mute your audio while attending the session. I request all the participants kindly mute your audio.
So can
And
I
is there?
continue?
Yes, sir.
Okay, so welcome each one of you to this talk. Today, the topic is actually computers and coronavirus. So there can be a lot of I would say that some misleading what the thoughts actually came to your mind. Sometimes, okay, what is going to be the topic or the content to be discussed on this particular title? Okay, so maybe it's not a purposefully put by me. Accidentally, actually, when I thought of what should be presented, accidentally this title came to me. Okay, anyway, because as Taslima pointed out, my research area is actually bioinformatics. So I thought of, okay, informatics anyway, it's there. So computers should be the maybe the because that is actually from where all the informatics actually starts. And now all of us are actually maybe knowing coronavirus, okay, that the term and how it works, and maybe the pandemic that is actually making a lot of trouble to the entire world. So that is actually the title actually came. Okay, so now we will move to maybe the coronavirus structure. I think most of you might have already seen these type of pictures while browsing for the last 6 months. Okay, so the left one is actually the a single coronavirus, and the maybe the infamous spike proteins that they are actually the yellow, okay, projections actually from the viral body. And okay, so the right one is actually the maybe a cross section, okay, where you can see the spike protein or S proteins, and inside you can see what is the viral genome. Okay, so here it is actually an RNA genome. So you can see that RNA and N protein. So that is actually the genome. Okay, we'll come to all this later, but this is actually maybe this particular viral molecule is the reason for maybe this particular say webinar series itself. So I thought of maybe starting with the coronavirus itself. Okay, fine. So that is actually about maybe the how a coronavirus will look like. So all these images and contents I have taken from internet. Okay, and okay, these are maybe some of the questions that might have come across with you. Okay, maybe at the time of your registering. Okay, what will be the contents that may be discussed, or whether this or that. Okay, so I thought of maybe when computers and coronavirus connects, there can be a lot of confusions or a lot of thoughts. Okay, that can come across with various people. So maybe the first one. So can coronavirus infect your computer? Okay, so that can be whether that is going to be the topic of today's talk. And so what can be the answer? Okay, logically you can say that directly it is almost impossible, because one is actually a living body and another is a non-living body, so there is no direct infection. Okay, but is it infect indirect? Okay, maybe not the computer system, but maybe say whether the commerce that is actually e-commerce that is actually happening, or the network that is actually where computers are connected, whether they can be infected by the coronavirus. So we'll come to that point later. Maybe another question can be, can computers help combat the coronavirus pandemic? Okay, So I think that there you may be telling yes, okay, because at least we are reading on the vaccine development and other things. So at least some of you might have come across with the articles or say web resources where you might have say read about this. Okay, if yes, then how the computers will help to combat the coronavirus pandemic? That's the second question. Okay, came to my mind. Then another one: can computer virus infect coronavirus and vice versa? So the first one is whether computer virus infecting coronavirus. I think may not be, but again, it's an impossible one. Again, the other way, that is coronavirus affecting computer virus. So that is also maybe illogical. But there is a very interesting thing. There can be maybe computer viruses. Okay, I think most of us know what is computer virus, and some of you may be knowing how a computer virus works and all those things. Okay, but there can be some inspiration, okay, biologically inspired algorithms you might have heard of. So whether any such inspiration the computer scientists, okay, can actually get from coronavirus, and maybe it's spread and it's working, which can be used to maybe improve the antivirus software. So that is actually maybe a point that can be discussed further. So that is actually another question. And the fourth one is actually, can computer viruses learn from coronavirus and vice versa? Okay, so that is actually again, so the first one, computer viruses learn from, that's a learning process, something like machine learning. Okay, or as we learn from the experiences, can computer viruses learn from coronavirus and vice versa? That can be again. And so can the mechanism of combating computer viruses apply to combating coronavirus? So that's a very interesting one. Okay, because we know how the computer scientists, or say the cryptography people, or the network security or computer security people, how they are actually developing very robust systems, okay, in order to combat computer viruses. So there are some standard procedure, okay, so steps involved actually in that particular computer viral defending, okay, or maybe finding out and taking further solutions. Okay, but can we apply the same techniques to coronavirus also in order to combat it? So these are some of the questions came to my mind. You can have other questions to add, and maybe you can look further. Okay. So this is actually maybe an article that I got from the Forbes magazine. So that's online spread of coronavirus viruses. Okay, it's actually how it is indirectly how a coronavirus can affect the spread of the coronavirus is an online manner. It's just it's a very catchy title they have actually given. So that means cyber criminals. Okay, so they are developing dangerous malware campaigns to target millions of users, because you know that nowadays, whenever you have a coronavirus or related things, but the terminology comes in a particular link or particular website, or wherever email or say WhatsApp attachment or whatever it is, people will anyway they will go and try to read it. So how can you utilize that particular point and maybe intrude into the computing systems and maybe spread the computer viruses? So that is actually the online spread of coronavirus viruses. Okay, so these attackers are sending malware-laced emails with the breaking coronavirus news. So everyone will try to, they will have a try to interest in, okay, following them maybe in the social networks. All these can lead to the spread of computer viruses. So new malicious websites related to coronavirus are also increasing day by day. Okay, so maybe it's a very perfect topic to, okay, hide behind the people and maybe do some malicious things. So that is actually very interesting actually. Okay, and now we'll go to maybe a little more detail on maybe how what is the similarity and maybe difference between this coronavirus and maybe computer viruses. Okay, because the topic is computers and coronavirus. So I'll be talking on okay, maybe on different angles, okay, how they are actually connected. So this is one point that we have discussed. And before going to the what is a virus and how that actually works with respect to coronavirus, okay, maybe we call it as a biological virus. Okay, so we should, I think all of you know about these very important molecules of life, DNA, RNA, and protein. Okay, so the DNA is usually it's a double-stranded molecule of made up of 4 nucleotides, A, T, C, G, and RNA is actually made up of A, C, G, U. Okay, and proteins, they are actually made up of amino acids. Okay, so there is it's a two-step process. It's a transcription followed by a translation. So that means you know that there is a term called a genome. I think you might have heard of, okay, the viral genome. Okay, so the genome is actually maybe the genetic makeup of an organism. Okay, so either DNA or RNA can be the genome of an say organism. Okay, but when the protein comes, okay, it is actually not a genome.
ഇപ്പോ എത്ര സെക്കൻഡാണ് അവർ പറഞ്ഞിരിക്കുന്നത്, അത് ചെയ്താൽ എത്ര പെർസെന്റേജ് വരെ ബാക്ടീരിയ.
Okay, so that is actually the protein. Okay, so that is actually called the central dogma of actually say molecular biology. And now we'll go to what is the similarity and maybe the dissimilarity that exists between say biological virus and a computer virus. Okay, even though there are a lot of difference, okay, between computer virus and biological virus, their working principle is almost the same. Okay, we know that viruses are already there, okay, from very long back. Okay, but computer viruses are actually human made. Okay, the programmers are actually written the code, and that actually resulted in computer viruses. So a computer virus, but making a computer virus and making a solution that is antivirus software, all these might be inspired by the working principle of biological viruses. So now we'll go to a little more detail about this one. So what can be the sample definitions for a computer virus and a virus? Virus, simply we say that a biological virus. So computer virus means can be defined as a piece of code. It's a program actually with a self-reproducing mechanism. Okay, so can reproduce itself riding on other programs because it need other programs to flourish. Okay, to work to maybe to launch it. Okay, and which cannot exist by itself. So the virus alone cannot actually exist itself. So see, the same way you can see that a virus, okay, it's a biological virus, we'll call it here, can be defined as a self-reproducing, okay, biological entity which uses a cellular mechanism and material to reproduce itself and cannot exist by itself. So you can see that a lot of similarity in the way actually they are actually working. So that is very interesting actually. Okay, I don't know whether we don't know actually whether this the person who actually wrote the first computer virus, okay, program, maybe might have studied, okay, looked into this biological virus and its working principle, and they have actually adopted that particular mechanism. So that we have to maybe go further and details, and no, it is not found such a relation. Anyway, it's very interesting actually. Okay. And this is actually a very life cycle of a biological virus. So I think you know that the biological virus cannot actually exist by itself. Okay, it is already present in the environment, but whenever there is a favorable environment comes, these viruses will get into the host cell. We call it as a host cell, and then it starts its action. So that is it's actually a very dangerous thing actually. So there are a lot of viruses in and around us. Okay, so when not in, it's actually around us. Okay, so whenever you have a virus got a favorable say environment, and it can come to the can attach with okay, it can bind with the host cell, okay, and using maybe some proteins, okay, which are actually say present on the, sometimes you call it as a receptor. Okay, there are different terminologies, okay, in the biological context. But essentially they will come and attach with the host cell, and maybe in our coronavirus, that is SARS-CoV-2 virus, okay, so it's an S protein. So that spike protein will help to come and bind with the host cell. That means maybe our cell, okay, and then it will infuse actually the genetic material that is present inside, okay. Usually in the case of coronavirus, it is an RNA virus. The genome is an RNA molecule, okay, and now they will actually come inside after the penetration. So first is attachment phase, then penetration. Maybe if you are actually knowing computer viruses, okay, you can just think from that angle, okay. So because you are actually, I think all of you are from engineering background, so you may be knowing what is actually the computer viruses. So there is an attachment, then penetration will come to that also. And now once it is actually inside the host cell, so what will happen is actually the membrane of that particular, okay, the protein coat, actually it's an actually coat, so that will break, and the virus will say release its genetic material, okay, to the host cell. And now this RNA to make a protein, okay, or DNA to make an RNA, you need a cell and environment. Inside the cell is actually essential. So now your viral genome is actually inside. And sometimes what will happen is that this virus will produce its own proteins from that RNA, or sometimes it's in a DNA virus, okay, sometimes it can come and fuse with our DNA, that means host cell DNA, which is actually present inside. Okay, now after that it will start its action, because it has coded for producing a few, say, proteins and other things. So what it will do is, it will create its own this blue protein, suppose it's a protein coat or so whatever it is. Now from that its own genome, it will produce a lot of proteins, and it will replicate, and it will create multiple or millions of copies or billions of copies, whatever it is, okay, inside the host cell. And now after some time, okay, when the size increases, it will come and assemble to a new protein. So that's very going to be again very interesting. So it will become a virus itself, a complete virus itself. And after that, as the size increases, this host cell is going to in a death, okay, and it will break the wall of this host cell, and this entire virus will come outside, okay. And the very another very the mechanism is that so when it comes outside, okay, through the host cell, okay, by breaking the its cell wall, it will actually get a lot of material, okay, or plasma membrane we call around this particular new viruses that are coming from the host cell. So that is actually going to be very challenging, because our other cells, which is actually working for the immunity, could not actually recognize whether this is a virus cell or it is actually a host cell itself. So this is actually life cycle of a biological virus. Okay, sorry. Maybe this is a piece of the code that I have taken from an internet source. So it's maybe a life cycle protocols such a hypothetical virus. Okay, so at the beginning it's actually one virus. Okay, and entry into the cell, injection of viral DNA or RNA into the host cell, transcription and replication mechanism, translation, assembly of progeny virus particles, exit from the cell, and at the end you have a lot of viruses. So that is the way a single virus can actually produce maybe thousands, even say millions of its replicas. So that's a very difficult thing to maybe solve. And this is actually how computer virus is actually maybe working, and some other related issues. So first one, you have to design, a programmer, okay, has to design. So developing virus code using programming languages or construction kits, whatever mechanism you are actually using, you are designing. Then it needs a replicate. Okay, so that means virus first replicates for a long period of time. You should enter into a target system, okay, either through maybe a pen drive or through an external device, or it can be through network, or it can be through any other way. It has to enter into your system, and it has to maybe launch. Okay, it gets activated with the user program certain action. So that means when you launch a particular application, the virus that is attached or that is programmed for that particular will also get launched. Okay, so once it is launched, okay, then it can actually maybe work like the previous virus, because if it is a your computer is connected to network, then it can go to, okay, inhibit or say it can destruct other computers which are actually present in the network. So that way it is actually the working principle. And you need detection, okay, fine. And once you detect it, then you have the antivirus software, okay, maybe just study the signature of the virus, and then, okay, to incorporate it, and then you have to install maybe the updates of the antivirus and further so that you can eliminate it. So that means when it is once it is infected, only you can actually identify the signature of the virus, and further attacks may be blocked by the antivirus. The first time it will be anyway infected, okay, that particular system, okay. So the same is actually happening with coronavirus or any other germs that actually say come to our body. So that's why once you are actually infected with any virus, okay, then your body will actually develop an immunity, okay, against that particular virus, and then it will actually maybe stop, okay, or it will defend against similar viruses, okay, in the future. So that is a similarity that exists. And okay, so this is maybe whatever we have discussed. You have on the left side, we have the coronavirus, and the right side, a piece of program. Okay, it's a very famous virus called Melissa virus. Okay, and so both of them are actually string of code, because it's a RNA molecule can be represented as a string of A, C, G, U letters. Okay, combinations in a particular way. The other thing is also a string. Okay, because it's a program. So in the coronavirus, it's RNA genome is a shell, while in Melissa, it's a computer code. So in both cases, the code is an instruction for the virus to follow. Okay, so that is a very interesting similarity that you can find. And this is again a comparison of biological virus and computer virus on different aspects. So one is actually, it is a parasitism. Okay. So the parasitism is actually very interesting, okay, because parasites, which is actually entering into, okay, a host cell, okay, and then using their resources for living. Okay. Then similarly, computer viruses are the same thing. I think what we have discussed. Then it is invisible phase. It replicates inside the organism for a while without any symptoms. Okay. When we get sick only, we will be knowing that, okay, there is a infection that has happened, but by that time there will be millions of copies of viruses in a second. Similarly, the computer virus also can be programmed to cause harm only after certain events, such as maybe launch of an app. So that, as we have discussed, okay. And what about mutation? You might have heard that the coronavirus is actually very quickly mutating. The one that is actually found in the Wuhan is actually different from that from US or from Italy or that from India. So there's a mutation rate is actually different. So that can produce copies with mutations. So what is a mutation is? The mutation means the change in the nucleotides, ACGU, in the RNA genome in the case of this coronavirus. So that means maybe a different protein, or say maybe a changed protein shape, okay, will be generated in a new variant. So that's a very complicated and very difficult to handle. So that's why this mutation is actually the reason for the vaccination is actually very difficult to produce. Okay, because you have to consider a lot of possibilities. And computer virus also can change the code in its copies to avoid a detection by antivirus software. So because you can program to change the code also. So that is again, there's a similarity, and there's a self-defense. Okay, so that means you can attack or comprehend. Our immune system can be beaten by this biological viruses. Similarly for computer viruses also. So that is actually some comparison. So if you would like to read more, okay, I have just found out a website. So if you go to that particular website, okay, I can see that the similarities and dissimilarities of computer viruses and biological viruses. It was first published in 2006, but it updated on June 2020. can see the significance of coronavirus. So you can see that a lot of similarities. Okay, computer viruses, biological viruses. Scroll down, there are 22 they have identified. I'm not going to the details. Potential similarities, dissimilarities. Okay, so a lot of things, a very interesting thing. Okay, because if you go through all these, maybe you can think from a research point of view, or maybe from some unique way, maybe something that some innovative solution or innovative problems that you can actually find out from this maybe this particular website. It's very interesting actually. Maybe natural evolution versus evolutionary algorithms. So you might have heard of evolutionary algorithms in computation. Okay, so all these are actually very interesting. So if you are interested, you can go and maybe further read it. Okay, so that is a detailed comparison. And another interesting study that has been done is actually propagation of the mode in which actually the maybe one cell divides into other cells, maybe the rate in which, maybe the pattern in which all these are actually say dividing and okay, maybe progressing. So I think again you might have seen that the engineers are actually very famous about the exponential curve, but maybe usually we mathematics class or in some other statistics class, you'd say, okay, there's an exponential curve. But nowadays you can see that we have seen this exponential curve of growth of this coronavirus maybe in daily, okay, in the newspaper. So it's a exponential curve is now everybody knows what is actually the shape. So you can see that the pattern is actually same for both computer viruses as well as biological viruses. Okay, this is an example. Okay, this is Ebola cases. Okay, that shows a trend, and this is actually a cod-red virus infection. And you can see that initially it will be very slow progressing, because one person is getting infected to 2, maybe 2 to 4, maybe 5, 6 initial period. But after that, after it reaches a particular number, then it is going to be exponential, exploding actually to very high number. Okay, so that is we might have heard of, say, flattening the curve and other things. Okay, so that is again, it's a very interesting. Okay, and maybe you might have heard of immune system. So what is actually immunity? Is actually how our body actually defends this particular viruses. Okay, so we have a built-in immunity system. Okay, you can see that it is a very, very complicated, but it is very efficient system. Okay, so but there are some limitations also. Okay, so our built-in immunity system is a self-enhancing, very interesting, efficient. Okay, it beats most of the germs. We know, because we get sick due to many fever, many other bacteria, other pathogens, but still we are actually living without any issue, because it will take some time, but after that we are again back to normalcy. So evolving, and but you have to practice some good healthy habits, because that's the only way you can actually get a better immune system. Okay, and if you look into antivirus software also, so you find the solution using research. Okay, because here and we have to updates needed against the latest viruses. That means that is also a learning process. Okay, it's also evolving. So you can see a lot of similarity that exists between an immune system as well as an antivirus software. And we know that in both the cases, it is prevention is a key. Okay, because if you can try to prevent it, then that's the best solution for preventing computer viruses as well as preventing coronavirus. That's why we are saying that stay home and stay safe, like that. But in computer virus, it is very difficult, because there you are preventing means you have to detach your computer from internet sometimes. Okay, or you don't connect any external devices. So, but in the case of computer viruses, it should be kept in a single room without any interaction, or there are no multiple users. Similarly, I think the quarantine is also something like that. So that means we don't have any interaction with others. You have to sit in a room and no other exchanges or whatever. So I think there is a similarity. Okay, so that is actually happening. Anyway, that is actually prevention is actually the key. Okay, and we should responsible behavior. That means whenever we are having some disease or some medical issues, then we should get a professional help. Okay, and we should not go to public places, and don't spread it. Okay, so there are some basic rules that everyone has to follow. Similarly, in the case of computer virus also, we need to have some responsibility. You have to scan the devices with the antivirus, okay, because unless you are not doing that, then there's a chance that the other system who are actually connected with or communicating, okay, they will be having a very robust and maybe antivirus system, but your system is not having, but there's a chance that it can happen. Okay, so that's very important. Okay, so that is maybe the first part where you I would like to or I wish to talk on computer viruses with the biological viruses. Okay, so now we'll go to maybe another angle. So I think again this is a very beautiful image that I have got from internet. Okay, so it is actually here you can see that the green are actually what is called cells, normal cells. Okay, and the purple color is actually the coronavirus particles. Okay, so you can see something like a cauliflower or something like that, because the spike proteins are actually seen there. So it is actually a picture, electron microscope, okay, electron scanning electron microscopic image of coronavirus particles infecting, okay, normal cells. So that is actually maybe another diagram. And now this is actually about, there are a lot of databases, okay, available for us to look into. Suppose if you would like to do a research in coronavirus, where I should go and find out the sequences or other details. So there is a virus called database called NCBA virus. It has been developed okay, recently and exclusively for this particular purpose. Okay, so you can see that I have given a search, and these are the results I got. You can see the first row. You can see the geolocation is actually India, Kerala state. So I just gave a with respect to India. So what will be the result that I will be getting? So okay. And you can see on the left side that Wuhan seafood market, pneumonia virus, okay, such details. Okay, and on the next page. When I clicked on the first link or the details, you can see that it is actually, you can see the attributes. It's a complete length. And what is the length of that RNA genome? It was 29,854. Okay, nucleotides, Homo sapiens, and the geolocation is India. Actually, the collection date is, you can see that 27th January 2020, which is, I think, the first infection that actually happened in India, in our say Thrissur district. So you can see, you can go and look at, and you will get the sequence. You have other information, and you can download the sequences and other information, and you can use this for your maybe research or for me study purposes. So that is another but from where you will get these details nowadays. And you know that say Wuhan was the first it was detected, and after that it maybe again it is actually human activity only actually spread that this particular virus through aeroplanes. Okay, because otherwise it would have been in China itself. Okay, anyway, passengers are there, millions of passengers going here and there, and that in that way coronavirus actually spread. Okay. So, and so each country has got their own researches and their research lab, and they have actually sequenced, okay, the genome, okay, from their own infected patients. So, and there is actually a consortium called GISAID, so who actually help the researchers to assimilate all these various sequenced genome sequences, so that researchers can actually use for further research like drug development or for say vaccine development. Okay, because there are so many variations actually happened due to mutation, and how it can be okay collected together and put in a single repository. Okay, so if you go to this particular website, you can see that it will take some time to load. So here what you will be seeing is that from the first corona, this SARS-CoV-2 virus or SCOV-19, whatever the terminology that we use, we can see all the sequences that have been deposited in this particular website, okay, with their details, and maybe their evolutionary relations are also displayed, or can study from that point of view. So there is a tree called a phylogenetic tree in bioinformatics, where it's a tree. I think for computer science people, you know what is a tree. You have a root and the internal nodes and the leaf nodes like that. So you can see that this shows, you can see that there are 4,868 genomes sampled between December 29th and August 2020. It's an updated one, okay? And so you can see that it actually started from this particular first one. This is a root node, okay? Maybe in the December, okay, 3rd, I think, okay, it is shown. And there was no much information for us on coronavirus on December 3rd. Maybe we might have read a small column in our newspaper that there is a virus outbreak in Wuhan. So, but things changed very quickly. Okay, so if you place your mouse on any one of these, okay, you can see the details, and if you click on, then you'll get those details. Okay, so that is this is called a phylogenetic tree. Okay, it's this particular tree is called a phylogenetic tree, and evolutionary how this viral genome got mutated, and who are actually closer, because this information will be vital for developing vaccines, because those who are closely related sequences will be able to get the same vaccine. Okay, but very distant means you need maybe sometimes a different vaccine or different say treatment itself. So it's a very crucial, okay. And you can see that the same information worldwide, okay, a lot of labels and other things. So I'm not going to the details, but still you have a lot of information, and you can see that these are the papers published with respect to this published genomes. Okay, so if you click on, then suppose I click on this one, and if you go back to your diagram, you can see that only one is actually highlighted. Okay, so this is actually the paper, the genome developed by this particular say group of researchers. So, and in that way also it can be analyzed for further research. So that is actually about so this particular website. Okay, and you know that it is a bioinformatics actually playing a very crucial role behind this COVID-19 vaccine development. I think you might, some of you might have already, or most of you maybe heard of this bioinformatics, and it is actually a robust application of information technology for acquisition, storage, visualization, management, a lot of things that you can, with respect to molecular biology data, that is actually the crucial part. Okay. So it's an multidisciplinary or interdisciplinary, whatever we call. Okay, so that is a very interesting thing. And there are a lot of subdomains under bioinformatics, biological databases like NCB virus that I have shown, and we'll have a visualization that's maybe we have seen example. Protein structures is very important. RNA second structure prediction, again crucial. All these are some of computer-aided drug discovery. That is actually where we are actually looking for some breakthrough, okay, for the coronavirus. Okay, so there are many domains. And this is just for example, what is alignment, because when you want to know how much a particular patient's coronavirus genome or say is related to maybe another person, or maybe in a nearby country or nearby continent, whatever it is, then you have to align these sequences, and you have to find out the similarity or dissimilarity, and so all these are, this is a particular area. It's very important, and there are so many computational algorithms, okay, like Needleman, Wunsch, Smith-Waterman algorithms, which actually help to find out this type of solutions. So that is a pairwise, 2 sequence at a time. But usually we have to analyze a multiple sequence alignment, because suppose you are having suppose in India there you want to you have a sequence from Kerala, and another sequence from say Delhi, and another patient sequence from say Uttarakhand, or another from say Kolkata, and you want to see whether any mutations or any changes happened with respect to the genome sequence, or that viral sequence. Then what you have to do is you have to do a multiple sequence alignment and see whether any changes or are they similar, so that they can, because the same sequences means there's a chance very high that similar proteins and similar working principle in the host cell. So this is again, you can see that this diagram I have taken from a paper on 2020, which has been published on COVID-19 genomes itself. Okay, it's a variant analysis of the COVID-19 genomes. So we can see that bioinformatics is actually playing a very crucial role behind all these development. And when you come to computer drug discovery, you know that developing a drug is actually a very challenging task because you have to identify the disease and you have to maybe isolate proteins involved in this particular diseases. That will take a lot of time, and then you have to test it to find out a drug molecule effectively. Okay, so which is maybe chemical compounds. You have to look into billions of molecules and see which molecule will be helping to maybe to work with this particular protein, and you need a preclinical testing. All these are actually the normal flow of the drug development. But using computers and using maybe the recent advances, like you say, supercomputing, machine learning, okay, artificial intelligence, whatever we call all these very advanced the buzzwords, or say maybe the new areas of computational computer science, you can see that all of them actually try to reduce the time that is actually required in each of these phases. So that's very important. So that's why we are expecting, or we are looking for, maybe machine learning will help, or say AI will help to solve, or supercomputer will help us to find out a drug or a vaccine, okay, maybe at the earliest. So that is the way research is actually going on. Okay. So that is again. So I think you might have again heard of this hydroxychloroquine. There's a very famous molecule, okay, because that was actually used for treating. Okay, I think India also exported a lot of molecules to other countries, and that is actually due to again a bioinformatics domain called drug repurposing research. That means the drug that has been used for a particular disease is applied on this patients also. So that's COVID patients also. So whether such things can be done. So again, you need a lot of analysis at the background. Okay, so because lot of molecules you have to, because there are again many drug molecules. So which molecule will be perfectly suited for this particular inhibiting this particular protein, okay, of this particular virus? Again, you need a lot of analysis has to be done, because you are applying this on a patient, so there should not be any the side effects. There will be side effects anyway, because most of the medicines that we use are actually having side effects. So that's another discussion itself, but still maybe in order to save lots of life, so can we use, even though there's a side effect which is not that crucial, then can we go ahead? So that way also the researchers actually advanced. Okay. And this is a way actually very interesting how our immune system works. Again, it's a bit of biology. Actually, because when a particular, say, germ or say pathogen, what we call like a coronavirus, enters into our body has got a two-layer defense mechanism, layer 1 and layer 2. So in layer 1, you have a say cells called macrophages and other things will grab the germ. Okay, and if they are actually a new germ or new pathogen, then there's a mechanism. Okay, so that means this macrophage, if they are non, okay, that means already infected one. You got a previous infection, then our body will straight away destroy them. But if it is a new one, then our system does not know whether it's our first layer, the soldiers. Okay, they don't know whether it's a new one, how to combat them. So now what it does mean, they will take the germ and it will show the germ's antigen. You might have heard of antigen test. That means whether there is an antigen is present in a cell or in a body. So that's antigen test. Okay, shows the germ's antigen to a cell called a T cell. So that is actually a cell, okay, present in our immune system. So what will happen once this germ is actually shown to T cells? Okay, this there are two types of T cells. One is called helper T cells, and another one called cytotoxic T cell. Okay, which will directly kill the germs. Okay, but what the helper T cell will do is it will take the germ and maybe the antigen, okay, and it will show to another cell called B cell. Okay, so what the B cell will do is B cell make a lot of copies, because B cell has got a memory cell. That's a very beauty of the, or the core of the our immune system. So that means B cell has got a memory, and whenever a new antigen is actually shown to this B cell will remember it, and it will make a thousands or even millions of copies of B cells to work against this particular antigen, and it actually creates the antibodies. Okay, I think you might have heard of the terminologies antibodies. So these antibodies are actually will go and then mark the germ for destruction, and other cells that will come and destroy this. So this is actually the way it's called an adaptive immune system, because we learn from, it's a learning process happening. Okay. And finally, when the next time this particular germ comes, then our macrophage itself will destroy them. So the second layer is actually the T cell B cell combination is actually the second defense, the first one. Okay, so that is how it works. Okay. Okay, so again, the fundamental premise is actually for the vaccine work, because in vaccine, these antibodies are actually inserted, okay, into our body. Okay, so that our B cells know, okay, this particular protein, okay, because we will inject this the antibodies to our body. That means the protein that has been maybe made in the body of the infected persons. So that if that is inserted to our body, okay, whenever the coronavirus infect us, okay, our B cells already know that whoever is actually coming from is actually the protein that has been already present there. So it knows how to, it has got that memory, so that it will go and combat without making us sick. So that's the way this vaccine is working actually. So we are not going to the details, but there are again, there are many ways in which the vaccine is actually developed. We are not going to the details, but strategies for a SARS-CoV-2 vaccine, there are many ways in which a vaccine can be developed. So one is actually live attenuated virus. So we are not going to the viral vector-based vaccine can be there, recombinant DNA vaccine, mRNA vaccine, lot of things. So this actually diagram again, same. So whenever you inject that particular viral antigen to this particular body, our body, then the T cell will activate, and the T cell will inform the B cell, and the B cell it will store in the memory also, so that next time it will combat it, and the B cell creates some plasma cell, and it creates antibodies. Antibodies will come and it will neutralize the viral. Okay, so that's the way it actually works whenever the vaccine will work. Okay. And I have also gone through how this AI and ML helped so far, okay, in SARS-CoV-2 research. Okay, so this is actually a table that I have got from the cited paper. You can see that ML and AI technology is applied in various forms or various problems in combating with the SARS-CoV-2. It is actually for screening. Okay, that means how screening can be done for patients. Okay, on what parameters, on what attributes. Okay, so how an AI or ML technique can be useful, okay, to make a better predictions. Okay, so that is a thing that you might have heard of deep convolutional neural network, support vector machines, random forest algorithm. All these have been applied. Okay, and the type of data they have taken, you can see that they, on which data they have actually applied. Okay, so this is actually one of the applications where AL and MI is already on. It's an ongoing research. And now you might have heard of about contact tracing app, something like our Arogya Setu. Okay, so these are actually a list of contact tracing app. So here also, I think some of you might have used Arogya Setu app. So it actually this type of contact tracing apps use your GPS or your Bluetooth or your some GPS or say other location based names, whatever it is, and all this information actually taken to the background, and then data also there, your call list and other things, they are also taken. Okay, all these are actually analyzed systems, and finally whether okay you are prone to a particular, have you contacted with a particular coronavirus patient, are you safe or not safe, all this information analysis can be given. So this again it's an application of ML, okay, or how ML is actually helping in this particular way. And this is another one, prediction and forecasting of SARS-CoV-2. So I think maybe suppose when in Wuhan, it actually came, or after that, say in Italy came, or Italy just got spread something like anything. Okay, then how that is going to affect, okay, it's in India, okay, so, or how that will affect in a particular country, okay, so that can be predicted actually, okay, maybe based on the mode in which it is actually spreading, maybe the social living style of a particular population, maybe the economic the status of the country. Lot of parameters can be taken into, okay, and social behavior, all this can be taken, and there are some studies who actually predicted how much, okay, maybe IIT Kanpur has predicted but I think in Kerala in September we will have 10,000 or even more cases per day. So we will see how much the predictions are actually correct. So all these are actually based on ML based techniques. Okay, so this is another one. I already mentioned that how supercomputers are actually can help us to fight this virus. Okay, so high performance computers are computing systems are essential, because you are not dealing with some say MBs or GBs of data. You are dealing with the terabytes and petabytes of data. Okay, if you consider all these things, so normally it is almost impossible to manually do, or maybe with the single systems. So you have to all of us or all the computing systems must come together and try to solve it. Otherwise, it is impossible to analyze this huge data. So based on this, the IBM and very big giants are actually, they have actually produced an HPC consortium for COVID-19 itself. That's a very interesting thing. So you can see that there are 50 there are 81 active projects of 43 members, and you can see that is 6.8 million CPU cores and 50k GPUs. So GPUs, I think, graphical processing units, all these are actually about increasing okay, maybe computational power, that means more people are joining, and all these are actually used. Okay, so if you can go to the site and you can see what is actually happening there, and maybe what are the solutions they have done so far and what is remaining. So you can see that who are the members, big giants, okay, Google, IBM, Amazon, everyone, because it is actually the problem of world. Okay, it's not a problem of one country or say one state or one company. You can see that MIT, okay, universities, top universities, all these are actually involved. Okay, and they're collectively actually say try to solve it. So you can see that 600 petaflops is actually the computational power there, CPU cores and GPUs, all these are actually combined together. So this is actually another interesting project. And quantum computers are also used, even though quantum computer is actually not realized, but still there are some principles applied to solve quantum machine learning. Okay, because there you need because which molecule is actually the best one to design a drug is actually nobody can predict, simply say. So you need different possible conformations of this particular molecules. So in order to make such variations, subtle variations, you need maybe quantum computer machine learning will be a better option than normal say computers. So that way also people, even at least in theory, because the pandemic can come maybe after a few years also. So at that time, whether the present results or research will be helping us to maybe quickly find a solution at that point. So in that way, it is actually again work is actually going on. And maybe the last slide, I think, folding at home. I think you might have again heard of this folding at home. It was actually initially used for finding extraterrestrial objects. Okay, but later it was actually used for used for maybe protein structure prediction, which is actually again a bioinformatics problem. I think after the extraterrestrial problem, then came the protein folding problem. Actually, folding at home is actually it's a distributed computing project for simulating protein dynamics, including the process of protein folding and the movement of proteins implicated in a variety of diseases. Because protein folding problem is considered as one of the toughest challenges, okay, the science has ever faced, because it's almost very difficult. Okay, maybe so. That's why but everybody is actually trying to solve this, okay, combinedly. So it brings together citizen scientists. Okay, citizen science, not anybody can actually contribute your computing power to this particular project. Okay, who volunteer to run simulations of protein dynamics on their personal computers. You know that usually we will be buying some 6 core, 8 core, or say 4 core or 8 core, sorry, machines with 2 CPUs. Okay, like that. But the question is, are we using the full computational power? Okay, very rare, very rare. Okay, even I don't think some 30%, more than 30%, no, your CPU usage will not be there for normal our day-to-day or even our research activities. Okay, our academic purpose. So why don't you give the remaining CPU power, okay, to this type of projects? So that is actually this particular project of folding at home. Okay, it insights from this data to helping scientists to better understand the biology and providing new opportunities. So this is a I will go to the previous slide. Folding it. So I'll come to. So this is a site. Okay. So here you can see again together we are say powerful. Okay, here also you can see you can download the folding at home application so that you can say contribute to this particular project, and you can see the progress on current COVID-19, some particular project to evaluate the potential drugs. Okay, it started on August 16th and it is completed 55.583%. It's progressing actually. So we can see a lot of things. Okay, so I'm not going to the details. So with that, I come to conclusion of say my talk. So thank you very much for your kind listening, and I hope it was communicated at least something, okay, beneficial for you. So thank you very much once again, IEEE Malabar section, for giving an opportunity. So thank you.
Thank you so much, sir. That was a wonderful and informative session, even in this pandemic coronavirus affected situation. Dear participants, if you are having any query, please drop into the either into the chat box or into you can ask directly to Dr. G. Gopakumar sir. If you want to directly ask the question, you can just hold your hand so that we can unmute you. Sir, my side, I'm having a question, sir. You were mentioning about different data sets available as part of this research work. So
Yes,
whether
yes.
it is we can download, whether it is we can accessible.
Yes, because all of them are public databases. Okay, so
Okay.
all the data is actually freely available for research purpose. No, because it's a part of NCBI, because NCBI is actually the largest consortium on biotechnology and bioinformatics. So they provide all the data under their website free for research. So you can easily download, because it has been there for the last 20-25 years.
Okay, sir. Sir, can you suggest any future research as part of that is the students can, they can work with some research along with this data set?
Yeah, the thing is that we may not be able to find a solution maybe by alone because we are not on the front line to have the latest data, but still this can be used for maybe in future, maybe prediction or maybe for developing but mathematical models that will help us to maybe in future. Okay, maybe especially for with respect to our state or say our country, how we can actually maybe develop some systems or models okay for such pandemic. So all those are actually going to be useful only. Even
Okay.
though in COVID-19 case we may not be able to give a solution okay very quickly because we will be getting the data after not the real-time data that we have, because they have actually found out this sequence and they might have done a lot of work, and then only they will be posting this data, okay, some of the groups.
Okay.
But still you can use this and you can develop a lot of problems. So for example, say prediction, forecasting. So forecasting is actually with what they have done is with respect to the current scenario. So anyway, now again forecasting in our Kerala is actually not a useful one because it's already ongoing. So now can suppose a particular similar type of virus that can come, okay, how the pattern in which that will actually spread maybe in India or in Kerala or another state, can we find out a model and how we can actually help to find a solution that will be useful.
Okay, sir. Fine. Any queries? I request all the participants to kindly fill the feedback form that we have posted here in this chat box so that you will be getting the certificate. Sabit sir? Hope there is. Okay, so there is no queries from the participant side. That was really a wonderful session, sir. So if we know more, many things about this coronavirus, about if you are hearing many things about the currently we are also going through many of the aspects with respect to this coronavirus. The session was simply super. Enjoyed it well, and all the participants might have enjoyed the session, and they might have got a very good information regarding the viruses and the coronaviruses. So thank you so much, sir, for being with us today for this webinar series, and hope we will be getting your support in the future also. You'll be there only. So thank you so much, sir, for being with us today.
Thank you very much for your kind words, and maybe yeah, I am happy. Thank you.
Thank you, sir. So participants, again, I'm requesting all to fill up the feedback form so that you will be getting the certificate. So with this, we are announcing that today's session has been closed. Thank you so much.
Thank you. So may I leave?
Yes, sir. Please.
Okay, thank you.